Done, sir.
Good. Okay, today we are very happy that we'll have a talk from Nagaland. This is the first talk from Nagaland, and that too in Nagamis. Nagamis, Nagaland laga local language ase. Aji Mukhan, Panchai discuss koribo, aru itu bi Nagamis te koribo. This is the first time that from Nagaland, Nagamis te talk hobo. So I request Asangla, or I request to please welcome Dr. Wati and the other people today. Asangla, please tell her to welcome.
Sir, can you please let her join?
Yeah, yes. 1 minute. सुनो, प्रथमेश मेरे को बनाओ एडमिन. प्रथमेश. प्रथमेश मेरे को एडमिन बनाओ. Vati, can you make her join? Vati?
Let me try.
Yeah, because you are the admin.
All right, sir. All right, sir. Is she online? I think that's the person.
Wati.
Yes.
Yeah, Dachin, no? Dachin.
Dachin
La from our lab.
All right, all right.
She's trying to join.
But I'm not getting any notification actually. Can you just request her to kindly try again?
Okay, okay.
I have the link ready, so I'll get the notification.
Okay, okay.
Yeah, she's online. She's online. Tachin is online.
Okay.
Okay, yeah. I joined. Is it on speaker?
Tachin, we can hear you. We can hear you. Just turn on your mic. We can hear you.
And you please welcome everybody.
Hello?
Yes.
Am I audible?
Yes, yes, you are.
Good evening, everyone. Good evening, everyone. I welcome you all for today's presentation entitled Exploring the Effect of Shifting Cultivation on Soil Rhizospheric Fungal Diversity in District, Nagaland. First of all, I would like to welcome Dr. Arvind Deshmukh, President of Microbiological Society India. He is an ex-head Department of Microbiology. Dr. Baba Sahib Ambedkar, Marathwada University, with 32 international journals and books, and 4 written and 7 edited books. He has organized 46 state, national, and international level conferences, seminar, workshops in India and abroad. I also like to welcome Professor Moranjit Singh, former head PNB Biotech CSIR MTech. He is listed in
Hello, can you please mute yourself? Can you please mute your mic? Yes, please.
Yes.
Yes.
Yeah, MTech director. He is listed in top 2% global scientists for 3 consecutive years. He is the director of SAS-free Polyclinic Perch, Mohali, Punjab. He did his postdoctorate from Pennsylvania Nias State University, USA, Kobe University, Japan, and Karl Street Institute of Technology, Germany. And I would also like to welcome our dearest ma'am, Dr. Dalijungla, panelist, is working as a professor in the Department of Botany and U, Lumami. She has published more than 60 research papers in journals of Nagaland and international repute, 9 book chapters, and 1 published book. I will also like to welcome our dearest speaker, Dr. Wati Demchen. Hello?
Hello, yes, please.
Yes. Dr. Wati Demchen, the speaker, he recently completed his PhD from Department of Botany, Nagaland University, under the supervision of Dr. Romeo Singh and co-supervisor, Dr. Dalijungla. And our panelist, Dr. Asangla Kichu, she has recently completed her PhD from the Department of Botany, Nagaland University, under Dr. Dalijungla. Yes, that's the introduction. Now I would like to give time to the speaker.
Yeah, thank you very much for the nice welcome. Itia moy Wati Temchen ke invite Korea se fungi te talk the bole. Please.
Very good evening to all the participants for this first presentation in Nagamis from our state of Nagaland. Presentation start noatamdi ni poila professor Swaran logo kushi kwa share guriaze taibra itu effort luigina ina platform dia garne mon sopra, not just me but the whole Department of Botany and under Nagaland University mon sopra bishi thanks ina poila kuisi before I start my presentation. As given in the topic, Nagamis ekalabra kweledo, there might be some communication gap, so Nagamis or English la mixture di kotarole try koribo. And if I go too fast or if I'm going too slow, anyone can just give me a feedback so that I make the best of time. Anyhow, I'll just start my presentation. So ajila topic to one shifting cultivation laga kota gule ase aro itu bra kina soil aro itu fungal diversity, especially itu rhizospheric mane root ti thakala region laga itu bra kina effect kuri ase. So mon sopra jani gina ase, we all know the cycle of shifting cultivation. We have a very large virgin forest area in the state of Nagaland. So mon bustimanora, what we will do is we will burn the area and we will sow the crop in this land. So this crop land, we call this the Jum land. So we will usually crop here for a period of 1 or 3 years. After that, one fallow land is Jabo, where basically the cultivated land is allowed to rest for a period of 10 to 20 years, which will allow the forest to recover. So itula face pishi important ase kile gole itu the soil recover kuribo aro nutrients consume tahibo. Besides the agricultural perspective, one shifting cultivation to especially in Nagaland context is very important. Why? Because festival species as with regards to shifting cultivation, for example, Auling Mungyu itu Kungya Kamra pre-harvest festival ikaze. Similarly, our tribe TV we have Tsongarmong, which is a pre-harvest festival, which is related with Juming. We also have the Tuluni, which is the biggest festival in the Sumi community. To festivals, identity, culture, pishi associated with these farming practices. So shifting cultivation in our state, it's not just a hand-to-mouth system, or it is not just purely an economic system, but the traditional institution there is a social bonding. It's very crucial for our survival, and since our region, Nagaland, it's on a hilltop, timber, forest, we get it all from Juming only. And itu brai moan, especially the farmers, the stakeholders, moan itu thakile moan nagamanoan bin nagamano ase ina, we get this sense of security. So it is very important from our perspective. And Guanchina itu thagara, because of this practice, moan we have this linkage between man and nature. So my work ita puribly, I would just like to introduce this work. It's the work done by Amba Ita. So why Nagaland? Money Nagaland dikile alternative shift practices I will took by, especially with regards to shifting cultivation, like alternative methods of farming. To Kwanishina, because of the unique geography of the district, that is the first thing, where basically because of this mountainous terrain, we cannot go for other farming practices. Another thing is the strong association of our nature and our socio-cultural practices, which is vital for our identity as Naga society. There are other practices, for example, terrace rice cultivation can be which can be used instead of shifting cultivation. But the problem is it will yield to be compared with this normal shifting cultivation. And lastly, there are also Ajikali government can scheme alag DSA with regards to cash crops or other plantation system. Will be for all these farming systems, the need of the hour is capital and financial resources and marketing infrastructure, which Nagalandi, it's very new, so we are a bit lacking behind on this. So these are the four main culprits that is preventing alternative means of farming besides shifting cultivation in the area. To Moan, itu keeping this point in view, Moan Nagaland la current situation to Saile, we find that Kohima, which is the state capital, almost 62% of the forest area is reported to be under degradation. Second, every year, almost 20,000 hectares of forest are cleared. Money for June cultivation karni, itu clear guna jaise. Another problem is because of the increase in cultivation, itu fallow period, which has been presented in the first slides, which is very important. Moan Sado, fallow period too, in some areas, especially in the eastern portion of the state, itu fallow period too, instead of the 10 to 20 years, it's in some cases, it is 4 to 6 years or even lesser. Another problem is one state, there's almost 100 different indigenous tribes, and it one sub indigenous tribe, they are completely dependent on Jum for their livelihood. And Kwanishina, there is a lack of alternative occupations for the indigenous inhabitant. Lastly, recently, what we have noticed is that there is practices of monocropping. We all know what monocropping is. So monocropping do fallow land ajikali practice great because of these economic reasons. So itan, when we implement all such practices in the area, ki hobo do soil erosion gan hobo. Soil erosion bra hobo, it will decrease the soil fertility. Idura plant la performance to be effect kuribo aro because of use of these pesticides and other chemicals, idu watershed siltation bi hobo. So ultimately, ki huado, there is a permanent change in the soil structure, which will lower the nutrients and microbes in the soil. Ultimately, idu soil organic matter content be decrease, which is very harmful. So ultimately, idura one la crop yield do iman negatively impact kuribo, which will threaten the livelihood security. So keeping all this point in view, our present work, what we have decided on was, since Mokokchung we have selected the district of Mokokchung, which has a very high population, which is dependent on shifting cultivation. I do experiment karni mohanba charta site chuise, which will be explained as follows. This is just the depiction of the Homotermic diagram for the study. So mohanba charta site choose kurise for this present study. So charta site to namely ekta Jumland hagiche, ekta do abandoned Jumland tree, the other one is abandoned Jumland bamboo, aro lastly abandoned Jumland 12. So it one site to explain guribu mira. So first Jumland wado tagan, the similar characteristic of all these four sites is that tagan sob, they have all been cultivated for a period of 3 years. The differences lie in the fact that first site, that is Jumlandi, fallow period thakanai. So it is directly going for the cassava monocropping. They have not implemented fallow period for this particular site. In the second site, it will be Jumland Hagiji will be itudo currently in its third year of fallow period money, farming practices stop purigna idia 3 sal hoise, and it is the natural vegetation as given in the table. The third site, that is abandoned dreamland bamboo. Itula site pi kwanishina farming 3 years karni thakije. After that, for 3 years there has been no farming activity. The difference between site 3 and site site AJL3 and AJLB is the difference here is that itula third site the farmers have actually planted bamboo sotulta, which has both economic reasons as well as it has been reported to increase the soil properties. And lastly, we have a normal fallow land, which is currently in its 12-year fallow period. So we are keeping the first site, that is jail, as a control to know whether the fallow period or the introduction of bamboo can impact the soil as well as the fungal components. So fungal colony kandu as discussed, rhizospheric collect kurishe, and we have isolated and cultured those in the potato textures agar and West Bengal. We have also calculated the percentage contribution to know how the fungal diversities are present in a different site. This is just a pictorial representation of the collection of the soil sample and its cultivation and subsequent pure culture isolation. Statistical analysis present study, we have gone for canonical correspondence analysis to know how exactly the soil, season, different soil parameters per kina rhizospheric fungal diversity impact kuri. What we've also gone further for is we have also examined the phosphate solubilizing fungi. PSF matya, these are fungi which can actually secrete some organic acid that lowers the pH, and ultimately this increases the phosphate solubilizing fungi. Nagaland soil is very low in nutrient. Nagaland is actually present in the hilly region, so naturally soil is very low in all these nutrients such as nitrogen, phosphorus, potassium. So the presence of such fungi such as PSF are very important with regards to yield. So present study PSF up to screen. These are the different soil parameters that we have discussed and that we have analyzed. We need to look at the soil parameters to know how these impact the fungal community. Soil data, it is very hard to know how the fungal communities will vary. So based on this, present work to so onto the result, as discussed, the first site which had no fallow period, we see that unlike the other 3 sites which were characterized by a clay loamy structure, clay or organic matter can be in the first site, that is jail, we find that clay can be it had a more characteristic of a sandy clay loamy structure. So we find from the get-go that the practice of reducing the fallow period has a significant impact on the soil structure. Secondly, this is the overall result of the annual soil collection. Notice kurado, when we compare the 4 sites, the main highlight that I would like to point out is the soil organic carbon. We find out that when we compare the 4 sites, the control site, that is site JL, site JL, there was no fallow period. We find that site JL soil parameters, so it was very weak. It was not very appropriate for growth, I dare say, as compared with the other sites. We see the best soil quality under AJL12, which, as mentioned, fallow period 12 years. So organic matter high because of all these different in the vegetation. Another interesting observation in the present report is that when we compare site AJL3 and AJLP, tuita last site P same fallow period 3 years. But the difference here is that AJLB, which had the bamboo introduced in the site, we find that it had very high organic carbon SOC as given in the table, as high as 3 as compared to AJL3. So we also see some of the remedial properties of bamboo with regards to the soil health. Besides that, if we compare all the other values, we find very significant differences between the different soil parameters, which shows without a shadow of a doubt that the different soil parameters are affected by the fallow land. So next work to what we tried to do was, when we present such data, raw data, if we present this data to the farmers, so it is very unproductive to present such raw data to the workers. So our next line of work was to convert this soil data into a very simple form, which can be farmer, ama, baba, so what we employed was the This is just explanation. I'll just skip this part. So Mohanbra Khutagura do bamboo la, which I've already discussed. Bamboo ra soil do help rekina do because of this very high growth of root formation. Because of this root, there is also greater mineralization process occurring inside the bamboo soil, and ultimately the soil organic carbon increase which favors the presence of the fungal community in the soil. So this is just a very simple illustration. What happens during Juming? So we have a normal vegetation soil with all its natural vegetation. The figure laga A. So whenever we commence this Juming, there is a high loss of carbon dioxide. When we do all those different anthropogenic disturbances, it basically strips the land of the organic matter. So when we introduce bamboo into this degraded Jum sites, because we bamboo can sequester carbon, they can capture carbon at a very fast rate. So the introduction of bamboo significantly increases the soil recovery processes. So as discussed, as I was discussing in the last slide, we wanted to incorporate a technique that will basically allow for easier information dissemination. So there are many methods available. Most appropriate technique for our present region soil quality index situation. That is the weighted method as per Rhizospheric et al. So what this method will do is it will collect the soil data. It will go through these dimension reduction processes. It will clear the data, clean the data, and it will give us a minimum data set from which we can build a soil quality index. So for example, instead of presenting the data in the tabular form, which is very hard, especially for people or especially for farmers, Purama, Babagan, Dalijungla, so if we can present the soil data in such a form, figure money value out of 10, it is very helpful. So we have converted our soil data into such a form, which will impact the stakeholders more significantly. This is just a pictorial representation of the different fungal diversity that we have collected from the 4 sites respectively. So last study lab purity, we have identified a total of 36 fungal species based on literature and based on expert consultation. What we find very interesting from this table, it presents the rhizospheric fungal diversity. So itu di what as microbiologists, we should always know what a particular fungi has a role, especially with regards to the agricultural perspective. To Mokokchung is the second largest producer of banana in the district. So in the state, so we all know Fusarium, it causes this banana wilt disease in plant. So last study, what we have actually noticed is there was this presence of this Fusarium in our present study. So if we go with the site-wise comparison, we find that inside JL, where there was no fallow period, we find this Fusarium species. In fact, there was 2 species. When we look at the AJL tree, that is the Jumland fallow period trees, there was a drastic decrease in the Fusarium species. Next, when we look at the AJLB, that is Jumland with fallow, Fusarium species in our 1 year of data collection, Olayolanai. Lastly, AJL12TB, similar studies. In retrospect, we find very high values of Trichoderma species, especially in AJLB and AJL12. So it will a relationship explore in the present study. Next to Monbra Sado, we have, as discussed, we went with the CCA, that is the canonical correspondence analysis, to know how the soil properties, how the seasonal variation, how it is all impacted by this. So what we find is, we find a very interesting observation. Monbra Siledo, in winter, we find that there is a very drastic decrease in the fungal diversity inside JL. As compared with the other sites. We also find that the maximum diversity was found during the spring and the summer in JL. Next, when we compare this same data with AJL3, we find that there was a more even distribution. The moment you introduce fallow, we find the distribution of fungal diversity to be more even as the season changes. A similar trend was also reported for AJLP. The introduction of bamboo also made the site very stable to such an extent that winter DB we could isolate around almost 10 fungal species. And lastly, AGL12, Mohanbra Kwanishina into AGL12, the itu subsea highest fallow period that is 12 years. Yadido, we find that yes, spring or summer the highest will be, even in winter, we could find very high values of this fungal community. Lastly, Mohan Kwanishina as discussed, itula present studied, we have also studied the presence of the phosphate solubilizing fungi. So in total, Monbra Atta fungal diversity by shape, which had the potential to solubilize fungi. If you look at the table, that is table 5, we find that under Zoomland, fungal diversity, the fungal population that could solubilize fungi, as compared with AGL3, which had 6, AGLP, which also had similarly 7, and AGL12, the maximum that is 7. So the picture this hello region indicates the presence of this phosphate solubilizing activity. Around Mohanbra, these 8 fungal species to Mohanbra, we have screened this. As per our study, we find that Aspergillus or Penicillium the maximum the fungi konbra phosphate to solubilize kure pare. Aro Penicillium laga highest solubilizing activity. So next we have gone for some indices just to confirm our finding. We find the Simpson's index of diversity was highest under AGL12. The higher the value, we find the higher the diversity richness. Similarly, evenness, we find the highest value of evenness for AGL12, which had the highest fallow period. This is actually reflected in our next finding, that is the Perger-Parker dominance index. So if the Perger-Parker's index value is high, it means basically that mane jagade there is a few particular species that dominates that particular area. So we find that under site JL, which had no fallow period, we find that that particular site had the highest Perger-Parker index, that is 0.12. So into a fungal population, it was very disproportionate. It was not even. So that also shows the impact of fallow and shifting cultivation on the fungal population as a whole. Next, as stated, Mohanbra Kwanishina itu even is to bishi kumdi thakije. Mohan kitia fallow period 2 sailedo. So Mohanbra bishi bakurize when Mohanbra kitia kheti kandu Mohanbra Jolabo, Jolaikana, salt can be spray guribo, fertilizers can alak thalibo. Itula time di automatically soil do artificial, we are making it very artificial, natural ishna hobo. So fungal species can adapt to the parle itula site. So mola present study bravi itu fact to aro ekbar Nagaland dibi itula trend to as reported by other workers. One similar observation ikta paise. Secondly, Mon kitia ina practices can continue kuriledo ki hobo geledo soil quality bi decrease kuribo. Itu mineralization can alak to fungal community or bacterial community which are present in the soil. Itu bra determine kurize. So whenever Mohan bra itu anthropogenic activities to increase kuribo, automatically we Nagaland the fungal population di Mohan bra decrease kuribo. Itu bra nutrient gan pona or nutrient mineralization process, nutrient availability so negatively impact kure. That is the first observation. Secondly, Mohan bra observe kurado fungal diversity increases when we when we incorporate into the fallow soil. There is a significant increase in the fungal properties. Aro kwanishina zooming di ajigale monoculture pishi hoise. After the zooming practices is complete, farmers can bra cassava, which is used as a livestock. Mohan ituan thaligina chaini charigina jaide. Instead of incorporating a proper fallow period, itu soil to recover kuruli napare. So itu be ekta problem ase. Itu stakeholders can highlight kuri lagi. Mane they are very much economic security karne down takan itu sob kurize holibi kitia ina chari dibo Mohan bra observe kuranishina fungal community do natural nishina recover nakuri. So ina hole do kuanishina kitia monoculture to practice kuribo, there will be some specific fungi which can adapt better to that particular plant. So itu last side to very disproportionate distribution of itu fungal species bra, it will dominate that particular site, which may lead to more pathogenic activity. So it ekta kotha dekhiche. Aro Mohan Kwanishina itiala present study, as per this present study, highest fungal species to Aspergillus aro Penicillium dekhiche. Itu kile kuile do itu species kan berdakan spore bishi punai, especially during isolation. So itu la reason ekta thakibo itu higher number dhadu in the present study. Moganbra Kwanishina, if we look back to the first figure, itu Fusarium species thakije, especially in JL. So it is a very big risk if this particular fungal strain is let loose, because we all know itu Panama wilt olavo, which will negatively impact the banana farmers, especially in our district. Kwanishina banana do it's very important. Manuansorpa Kaiginabi, there is a very high economic importance of this particular plant. As stated, Mokokchung it is the second largest producer of banana in this district. So Moanbra itu practices, especially monocropping and palbrana sile, this fungal strain may also impact the banana productivity under the site. Aro Kwanishina fallow period kina introduce kuribo, aro bamboo gan kina introduce kuribo, Moanla study di pado Trichoderma last species to increase kure. So kina gure goledo itu this Trichoderma as a fungal species has some antagonistic impact on the Fusarium species. So mui ekta paper puri thagishe where basically itu Trichoderma Harzineum Z551 strain bra Fusarium oxosporium do palbra regulate kule pariche. So that if figure ekta diase when you introduce the Trichoderma species in the presence of Fusarium, takan brabi report be look similar by mane. The more you increase the Trichoderma population in a particular soil site, the lesser the Fusarium species are found. So such the biocontrol, such biological agent, it one state they should be definitely screened out for better utilization in the district. Aru Kwanishina shifting cultivation do kitya itu palra inan hobo Kwanishina diseases can spread kuribo in the site. It will also negatively impact the biodiversity of the region. So another finding of the study, Kwanishina, there was a very high fungal diversity in the bamboo site. So Kwanishina, bamboo ganbra, as we all know, these are very rapidly growing grass. Because of their rapid growth, the carbon species sequestration, it makes the soil very rich in carbon, and ultimately fungal diversity can be increased kuribo. Bamboo bra, it has this capacity to produce very large number of fine roots, which will basically increase the surface area for mineralization processes. Another finding is that in Jumland, another problem is, as per the canonical correspondence analysis, Moanbar report pado winter the fungal species pishi kumtiway. So in such drastic changes may prevent the site from recovering its natural fungal population. So in a problem there might be actually not just a disappearance but a permanent loss of such fungal strain in Jumland if this practice continues. Arugwanishina Moanla present study be not just the other fungal species but itu phosphate solubilizing fungi can be pishi high, especially in the AGL bin AGL 12. So we can also conclusively state that when we increase the fallow period, or when we increase the introduction of these such plant species that can regenerate the plant properties, it is not just the soil quality, but we are also giving opportunity to one phosphate solubilizing fungi or there are many other varieties of fungi. So it one opportunities for them to recover, for them to basically come to more efficient. So in a wado, secondly, maximum fungal species to Aspergillus or Penicillium thakije. So I think in the future it will the point to rakhigna, it will data to baseline data nishina rakhigna, future screening or future pod experiment kan jaile bishi bhalabo. Kile koile mon Nagaland we are looking we are aiming to be a very organic state with regards to our agricultural production, though in naga biological agent thakigna which will cause no harm to the environment. In a methods kind of implement kurna large scale field experiment gurle pishi bhalo ina ekta suggestion ase. So in the last conclusion, the main takeaway is that whenever you negatively impact the soil quality, one Kwanishina fallow period to reduce or increased level of anthropogenic disturbances soil quality to biya hobli ase itu ta kota. Secondly, bamboo gando thalile bhalo hobo in Zoomland. There are two advantages here. Firstly, soil recovery bishi bhalo hobo. Secondly, bamboo laga price to Mokokchung moi aji huta do 150 rupees as a per piece. So farmers can lobby instead of just leaving the forest bare and just waiting for 10 to 20 years for it to soil recover kurubule. Instead of that, they can sell the bamboo. Meanwhile, automatically they're getting both economic return as well as soil health. So it's a win-win. Secondly, land use Tikwanishina, there is very high impact on the fungal population. To Muikwanishina Muan Musa caliber, that is banana. Since Mokokchungdi, it has a very high dependence on banana. Itugan la data kan di phala monitor kurle phala ba. Noile inaga practices continue kurle, high chances that the fusarium wilt, which is not so prominent in the district, prominent hobole pishi chances ase. So such practices, such information, villagers, farmers, stakeholders kan lo, department kan lo diguli lagi. Lastly, Kwanishina, the presence of these microbes alak alak kan lo idi pishi phalaje. Idu especially those fungus which can solubilize fungi. Itu one thagili pishi bhalo bo. To stakeholders, researchers kan aro the experts can also definitely look further into this stream. Inna jailedu Kwanishina, our road step to becoming an organic state pishi bhalo bo inna bhabize. So in conclusion, these are just idu references kolop thagije mula presentation la. Aru thank you for this time and opportunity again. And if there's any question, I will try my best to answer it to the best of my abilities. Thank you again.
Thank you, Dr. MT.
Thank you.
Oh, Vati.
Congratulations.
Very nice presentation.
अपनी दिसे भाल टॉक दिसे अपनी वाती टेंशन मैं एकदम भाल लगी से I came to know that Nagaland has got such a good soil because of bamboos, and now even coffee plantation is there in Nagaland, right?
Yes sir, yes sir.
So it will अपने का टॉक पर बिसी भाल facts मैं खान के पैसे हाँ आरु मैं खान तो जाना नहीं पहले तो जाना नहीं इतने जानी हाँ. Now I request the panelists Dr. Dalijungla and Atangla to ask a few questions. Dr. Dalijungla.
Yes, sir. Yes, sir. I congratulate Dr. Wati for the excellent presentation.
Thank you, ma'am.
And a very updated one. Actually, yeah. Just not the question, but I would just like to supplement one thing. Bamboo plantation, we do encourage, which has very high economic value, but we don't encourage the farmers to go for just bamboo alone. Along with the bamboos, especially the Bambusa dolda, along with that, the timber yielding plants and other crops as well. What happens if we just do the bamboo plantation alone in a plot? Then slowly, other plants you will not allow to grow in that bamboo grove. So that is one thing. And whatever fungi, Dr. Wati, you have identified, whether you have done any molecular characterization?
Actually, ma'am, we could not go with any molecular analysis. The main thing is we could just identify till the species level. The main problem with our this thing was firstly was the pandemic. We could that was the main thing. Another problem was that most of our fungal strain, because of our lab facilities, we kept it there, but because of this pandemic, one jawli paranai. So it will be a lot problem thagiche, but definitely looking forward to go for more molecular analysis in the future.
itu lagado we mokan 18 months nishna eku lab de kam koribole para nai itu bra bi time busy waste hoise, and soon after we came back from pandemic, I mean the lockdown, now the time limit because the scholar has to finish within a stipulated number of years. So that could be one of the reasons why we would not do up to the molecular characterization for authentication of our fungal species. So you see Bhalase, more members
Very
can
good.
come discuss.
Nice presentation. Can I ask something?
Yes, sir. Yes, sir. Definitely.
Yes.
Okay, Vati, I congratulate अपने काम तो बेसी डांगोर कोता को ऐसे बेसी बाल काम को ऐसे और वो मोई तो बेसी खुशी पाए से इमेन डांगोर काम अपने लुमामी ते थाका पीछे ते कुरे से मोई बेसी खुशी पाए से और वो एकटा मोई सजेशन और क्वेश्चन भी करे वो लगे तो इतो पर आप मोई अपने इनका ट्राइकोडर्मा रिपोर्ट करी से और वो अपने तो जाने वो लगे ट्राइकोडर्मा तो वेरी बेनिफिशियल फॉर दी प्लांट्स सो इफ वी यूज वर्मी कंपोस्ट along with trichoderma I think that will help the crops even the soil also even the soil also they will help
Definitely.
So that is the one thing you encourage the culture of trichoderma, mix it with the vermicompost, that will be double benefited.
Definitely, sir. Definitely, sir.
And other thing, other एकटा कोता को बोले मना से ये तो अपने बैम्बू लगा बेसी एडवर्टाइज करे से like in Nagamese I'm saying so डांगोर कोता को इसे बैम्बू कल्टीवेशन लगा किंतु मुझे अपना बोटेनिकल गार्डन आसे ना
Yes, sir. Yes, sir.
मुझे एस्टेब्लिश करे से ताते तो बेसी बैम्बू आसे
Yes, sir.
ताते तो मुझे ना बेसी टर्माइट पाए आसे बैम्बू रूट ते तो टर्माइट बेसी ग्रो करे तो इतो भी एकटा मनते कोता रखिबो लगे
Yes, sir. Yes, sir.
इतो टर्माइट बेसी ना उलाइले तो भालो हो ना
Yes, sir. Yes, sir.
ताई तो क्रॉप ते बेसी नुकसान करे ना तो इतो भी किंतु मोय मन तो बेसी खुशी अपने बेसी भाल काम करे से when I was there you are working in some project on banana
sir. Yes, sir. Yes, sir.
I think this study is very nice.
Okay.
Very informative, good information, and this is I think whatever you are reporting that is first hand study, na?
Yes, sir. Yes, sir.
So I'm happy. Congratulations again.
Thank you,
Keep
sir.
it. Now what are you doing nowadays?
Thank you.
Sir, I've just finished my PhD last month. It's been 1 week, so
Okay. So all the best for your future endeavor.
Thank you, sir. Thank you, sir.
Good luck.
Thank you, sir.
And to your
Can I call upon
supervisor also, congratulations.
Thank you, sir.
Dalijungla. Right. Okay, can I call upon you or discuss something? Yes.
Yes, Wati, it was such a wonderful presentation. It was so informative. Thank you. Wati, I just want to ask a question. Abdi itu bamboo plantation itu bara soil nutrients can increase kuride na the presentation that you have mentioned, right? So apart from that, itu monocropping, how does it affect the fungal population? Itu can you please explain?
Oh, thank you. That's a very interesting question. Actually, kwa nishina fungal community to takan kitya soil di dhabo. They have a specific preference for a particular plant. So the dangers associated with monocropping in general, though, because ajikalin one we've all heard, Nagaland we are about to start our coffee plantation ka monocropping. So kitya ina hobo, what happens is those particular fungi that can adapt to that particular plant tan dominate hobo. So kwan nishina, for example, moan la present study moan brad dikhado kitia monocropping, for example, cassava di puribo fusarium species to pishi hoize because itula particular species though it can drive there basically. So it has a particular pathogenic activity that can basically diminish the population of the other fungal species. So I think what we should all do is itia I'm sure lab kandibi we have seen some of our researchers working on this, but it's very important that we look at how this coffee plantation in particular itu rabi kina fungal community to impact puribo because one ishina coffee plantation for the next 4, 5 or even more years we gina what if there is a presence of a particular strain of fungal species that not just reduces the yield but it can also significantly just damage the whole crop. In that hagilido is very dangerous. So monocropping lado we all know bishi dangerous species so itya since we are at the pioneering stages itya this is just a start. Based on my data and even based on our future data, it's very important to know how the monocropping of this, especially coffee, will influence the fungal community.
Okay, thank you.
Very good. Okay, what's the time, Jen?
Hello, sir.
Zooming laga. Hello, zooming laga ki effect ase fungus.
Actually, sir, inaze zooming directly. It's not actually directly because inaze when you burn the soil, zooming la first step to you have to burn the soil. So soil ekla burn gurabrado in 30 days or even in 1 month, the fungal community can recover. Itulado ko problem na ide. The fire is not the problem. But kwa nishina, the practices of zooming, ito one key se we are depriving the soil of its natural vegetation. Though obviously ito kota do fungal community natural fungal population na thakibo because for example if we bring rice laga we will get a different variety of fungal population. So ito ekta kota. Next to we are increasing the cropping phase. So disturbances, anthropogenic disturbances, for example, salt, nimok can use in the zooming sites to control weeds. It is directly impacting the soil. So ultimately, there is a permanent change in the soil structure, which will basically determine the fungal community. So Kwanishina fallow period to Kwanishina Bishop Halaze, but it's a very slow recovery phase, 10 to 20 years. And we cannot just tell the farmer, you just wait for 10 to 20 years. So Kwanishina, we need some restorative potential, some agents, biological agents, as mentioned by our madam. Bamboo, it is not the only solution, but it is among one of the many solutions. Though I think as researchers, we have to look into all this and just proceed further, sir.
Zooming bacteria ke bhi mari di. Khali fungus ke na mare.
No, bacteria bhi sir, definitely even bacteria. Morai dibo.
So bacteria, can they revive very soon like fungus or not?
Actually, sir, inaze, bacteria, as per literature, bacteria can recover faster. But the problem is because of their very high rate of recoverability, fungus though it takes some time because they have to produce all those spores and this thing. But bacteria though, what will happen is ekta kota tuita bacteria thakise. Aro itula recovery period lo faster thakile though this will basically dominate the entire area. So itula problematic with regards to bacteria.
That's right. Actually, bacteria is a function of the bacteria. So plasmid, the bacteria can generate very fast
sir.
comparing to
yes sir.
So it's the last question मैं आपने के दिवो. Soil monitoring.
Yes sir.
Soil monitoring लगा cost. मैं खान किने का कौन करिबो पारे.
Sir, that is a very good question, sir. Actually, Soil monitoring is very expensive. I'll be very honest with you. Very expensive as a lab tea, reagent gun, or the amount of work that is required to monitor the soil. It is a very large piece of land. Though, Kwan Nishina, direct Puila Nishina, just for a descriptive study, it will cost.
Yes.
It's so expensive. Aro Kwan Nishina, we can also go to soil laboratories, but there is a certain cost attached with all those monitoring. So Moi laga review la sab tido, and even in my thesis, mohan bra ki kura do mohan bra soil quality index to introduce kurise. So there are two advantages. So we all know this term correlation. Mane ekta change kora nande ekta kina change kore. There is this positive correlation and negative correlation. So the good thing about soil is that the soil nutrients, their texture, they are not independent of one another. They are very much dependent. For example, if you increase the soil organic carbon, conductivity will increase, nutrient will increase. So, ito correlation dhagado, what we can actually do is, if we can implement some statistical technique that can isolate some particular soil property, instead of monitoring 20 soil parameters, we can just monitor 2 parameters. Ito rani soil health to kobole pare. So, mogan present study di mogan bra do weighted method soil quality index connect aze. So, it will capture the data. And another advantage is, instead of presenting the raw data, for example, mui dia pusi di jagna mui la grandfather logo, tu la feel the phosphorus level 4.6 kg per hectare is able tai kun na buju boido bra. Tai ni head scratch koribo. Though soil quality index la phalekta do, it will convert the raw data into a tabular or into a figure. Aro taka do paribo. Like apni la soil inaze 6 as a 7 as a 10 as a to it's very easy to this thing. So I believe from our state Moibra first with regards to shifting cultivation, our present work which has already been published, that is our first report of implementing this soil quality index in the region of Nagaland. So I believe, sir, that is one among the many techniques that can reduce the cost as well as the work required for soil monitoring, sir.
Very good.
Thank you. Very nice.
Thank you, sir.
I can see Dr. Zubon. Dr. Zubon?
Hello, hello.
Hello, sir.
How are you? How are you?
I'm good. I'm good.
Very good. So you want to ask something to Wati Temjan?
No, but I just came now. It's just like 1 minute only.
Okay.
So
No problem.
I'm not in the part of discussion.
Try
So
to
sorry.
talk this fungus report. So any more questions by anybody? Any more questions? Okay, come on. Does somebody want to ask a question?
Sorry, sir. I think I have to leave the thing. We have one more emergency surgery. I'll come back later.
Okay, bye. Yeah. So if there is no question, then I think we can have the vote of thanks.
Okay. Good evening, everyone, once again. It has been such an honor to be a part of this wonderful event. On behalf of the Microbiological Society team, I would like to extend my appreciation to our distinguished speaker, Dr. Wati Temjan, for not only sparing his invaluable time, but also for enlightening us on how fungal and soil but also enlightening us with the relation, like how the fungal and the soil has on their relationship, and how these observations were helpful for soil fertility and by the experiments which were condensed for the layman to understand. I would also like to extend my heartfelt appreciation to Dr. Arvind Deshmukh, President of Microbiological Society India, and Dr. Swaranjit Singh, Principal Investigator at Institute of Microbial Technology. I would also like to express my sincere thanks to the panelists and participants for their enormous cooperation for the event. The event was very enlightening, as well as it was valuable for all the members present here. And we all look forward to this kind of event again. Thank you, one and all. God bless you all.
Thank you very much for the nice welcome. And now the vote of thanks. Thank you very much. Bye.
Yeah, thank you. Bye.